Hello and welcome to our last lecture, formal lecture for this semester in drugs and human behavior. Today I'm going to talk about pharmacogenetics and pharmacogenomics. First, a little personal disclosure, this is not my area of expertise. It's an area that I find fascinating. There are lots of really great resources on the web if this is something you're interested in. There's some great textbooks out. Be sure to look at those. There's a really terrific article in the British Journal of Pain from the British Pain Society by Craig et al. Called Pharmacogenetics of Analgesic Drugs. That came out in 2013. A good deal of what I'm going to talk about came from this and other peer-reviewed articles available from the National Institute of Drug Abuse or the Substance Abuse Mental Health Services Administration. So today we're going to talk about genetics and drugs. We'll start first by providing an introduction to genetic expression. We'll talk about epigenetics in drug abuse, and then we'll finish up with some specific discussions, albeit kind of brief, on pharmacogenetics and psychopharmacology, in particular focusing on most of the discussion on genetic differences in the cytochrome P450 metabolism enzymes, and some genetic variations and how those are expressed. But let's start with genetic expression. As most of you should know, the primary function of DNA is in creating proteins, or in protein synthesis, and this is accomplished through transcription. Information stored in a gene's DNA is transferred to messenger RNA. The messenger RNA then is able to transcribe the information from a gene's DNA and then translate that into protein. So messenger RNA interacts with the ribosome, which reads the sequence of the mRNA bases. Each sequence of 3 bases, which is called a codon, usually codes for an amino acid. Transfer RNA, or tRNA, then assembles that protein 1 amino acid at a time. Protein assembly continues until the ribosome encounters a stop codon. So this process is the way in which our DNA tells our cells essentially what proteins to make and essentially what to do and what to be. It's a very simple introduction to this process, but these are the processes we're going to talk about in terms of how drugs of use and abuse can affect this process through epigenetic alterations, and also, of course, how alterations in the genetic code itself can change protein production of things like enzymes, and we'll talk about then how that affects drug metabolism. So one of the biggest things that happens is genes can get turned on and off. The process of turning genes on and off is known as gene regulation. And gene regulation can occur at any point during gene expression, but most commonly occurs at the level of transcription. One of the things we know happens is our experiences can turn genes on and off. So, for example, human beings are one of the few species who are capable of digesting dairy products after we have been weaned from our mothers. Most species don't consume dairy. In fact, most species can't consume dairy. At some point in our evolution, that gene remained switched on, and that occurred after we domesticated cattle and goats and started consuming dairy and cheese and whatnot. Now, in many people, that gene switches off. These are, of course, people who are lactose intolerant because they cannot produce an enzyme to break down lactose in milk. And that's really normal in the evolutionary process, but we've been able to sort of keep that gene switched on. Similarly, we have to manufacture a specific enzyme to digest beef protein. Some of us can't produce that, and as a result, can no longer digest beef either. So, the next thing we'll talk about in our introduction to genetic expression is talk about epigenetics and the epigenome. The epigenome compromises all the chemical compounds that have been added to one's genome as a way to regulate the expression of all genes within a genome, which is a lot of words. But basically what happens is we're born with our pure genome, as it were, supposedly, and then as a result of all of our life's experiences, the colds we've been exposed to, the times that we injured ourselves, the all the things that might have happened, stress, poverty, violence, stress, drug abuse, anything, all of these things alter the way in which our genes have been expressed. So epigenomic modifications remain as cells divide and in some cases can be inherited through the generations. So the alterations caused to a genome in one generation can then influence the behavior of the next generation. And we're just at the very beginnings of understanding this process, but it's really important that we get a handle on some things in order to try to understand what's happening with the next generation. So epigenetic changes can help determine whether genes are turned on or off and can influence the production of proteins in certain cells. A common type of epigenomic modification is called methylation. When methyl groups are added to a particular gene, that gene is turned off or silenced and no protein is produced from that gene. So this is a common way in which genetic expression is changed by the alteration of the production of that particular gene. So to give you an example, epigenetics could explain why some people suddenly become lactose intolerant because they're no longer producing an enzyme, because that methyl group was added to a particular gene that turns off the production of that particular protein. So these kinds of genetic modifications can have influences over how we process foods, how we process drugs, how our brain handles drugs. Anything involving the production of proteins, including things like neurotransmitters, can be influenced. So I want to talk a little bit about genetics and psychopharmacology. First, talking about pharmacogenetics. In general, pharmacogenetic usually refers to how variations in one single gene influences the response to a single drug. This may be useful for targeting appropriate treatments. Pharmacogenomics is a broader term which studies how all of the genes, that is the genome, can influence responses to drugs. Where much of the research looking for target genes occurs is in pharmacogenomics. The terms are often used interchangeably. I think pharmacogenetics is probably most appropriate for what we'll be talking about, because usually we're talking about something very specific altering in a person's genetic structure. So we're not talking about the entire genome, we're talking about a very specific part of the genetic structure or so of the genome, so genetics. So we'll talk about people who are poor metabolizers of drugs or ultra metabolizers of drugs because they have alterations in the proteins that create cytochrome P2DG. One of those, cytochrome P450, enzymes produced that break down certain drugs or metabolize certain drugs. The last area we'll of course talk about is epigenetics, and we're going to spend some time talking about epigenetics of drug abuse, and of course we just introduced this idea of how epigenetics works. So let's turn now and talk about epigenetics of drugs of abuse. We'll first talk about genetic predispositions to addiction, talk about environmental effects that may regulate predisposition to addiction, introduction to epigenetics, and we'll talk about epigenetic alterations caused by drug abuse and addiction. So we'll start with genetic predispositions to addiction. First of all, twin studies have shown that there are common heritable components to drug addiction. Genetic factors contribute to 25 to 50% of the variance of developing a drug addiction. The A1 allele of the dopamine receptor gene DRD2 is more common in people addicted to alcohol or cocaine. And non-smokers are more likely than smokers to carry a protective allele of the cytochrome P2A6 gene, which causes them to feel nausea and dizziness from smoking. So these are just a few of the genetic properties that may or may not contribute to our propensity to addiction. So again, twin studies show that there are common heritable genetic components. There is estimates of about 20 to 50% of the chances of developing a drug addiction appear to be genetic. And this is one of the important things I think it's really important to stress, to have a truly nuanced view of drug addiction, is we're not talking about people who have made bad decisions. We're talking about people who are at risk from the time they might have started a drug. So if we can identify those people earlier on, we might have a much greater chance of dealing with drug addiction rather than simply having a one-size-fits-all approach. And I think that's really important. So again, we have different alleles in the dopamine receptor genes that are different in people addicted to alcohol or cocaine. And again, we've already talked a little bit about this, but genetic alterations in the propensity for smoking is another one. Environmental effects may regulate predisposition to addiction. Environmental conditions can regulate or fully attenuate genetic predisposition to addiction. So while there is a genetic predisposition to addiction, some environmental influences can either mitigate or exacerbate those predispositions. So in adolescents with a short 5-HTT-LPR allele associated with serotonin transporter expression and risky behavior, exposure to supportive parenting or membership in community building initiatives reduce substance abuse initiation. So this is a really unique idea where we can identify a specific genotype or phenotype with disruptions in this serotonin transporter expression and is also associated with risky behaviors. One of the important things to understand about some of this genetics and behavior research is you have to try to associate specific genetic traits with behaviors, and it's always a bit correlational, but I think we're starting to build a much better understanding of this. This is a really young field, but a really exciting one, and so I hope some of you will continue on to look into this area. Stress then can cause methylation of genes, which alters our ability to deal with stress. So that's an environmental effect that can increase our potential for addiction. We know that people who are under a great deal of stress, who are in high stress occupations or in high stress situations, are at much greater risk for addiction, and that seems to be potentially caused by alterations in their epigenetic structures. Again, social support can be a moderator of this effect, so good social support can moderate the effects of risk for addiction, and I think that's a really important thing for us to understand. So I want to provide a little bit more thorough introduction to epigenetics, particularly to how these structures get altered. So we talked a little bit about DNA methylation. Another process was what's called chromatin modifications. These are linked processes. DNA methylation may be a signal to package DNA in a closed chromatin formation. Changes in DNA methylation may contribute to memory consolidation and synaptic plasticity, processes that we know might be associated with addiction. Addiction, after all, is a learned process. It's the brain has learned to associate drugs with specific outcomes. Then the modulation of gene expression that mediates drug-induced neuroplasticity have been found to be related to learning and memory and may be related to learned drug habits. So we do have very good reasons to believe that epigenetic alterations can be associated with drug addiction. Again, alterations in gene expression may be related to relapse. We also know that administration of drugs of abuse can trigger epigenetic alterations that regulate transcription, that these changes may influence reward, psychomotor activity, drug craving, and relapse. And then finally, responses to pharmacotherapy for a treatment of abuse may be determined in part by epigenetic factors. So trying to determine the best treatment for someone who is addicted to alcohol or drugs may involve determining some of their genetic structures, some of their genetic coding, and then try to figure out some targeted treatments for those people. So the final thing I want to talk about under epigenetics is to talk about epigenetics and alterations caused by drug abuse and addiction. In particular, we're going to talk about cocaine and opioids because it seems to be where we have the greatest deal of information on how these drugs might cause epigenetic alterations. So behavioral addiction and neuronal plasticity are rooted, in part, in epigenetic mechanisms. So getting addicted to a substance and changes in brain functioning or neuronal plasticity are, in fact, rooted in part in epigenetic mechanisms. So I want to talk first a little bit about cocaine. There are immediate and lasting epigenetic modifications from cocaine use. In the nucleus accumbens, a single injection will rapidly increase early genetic expression of C-FOS. C-FOS has been associated with a variety of addictive processes. Prior long-term exposure to nicotine may induce epigenetic alterations with heightened sensitivity to cocaine and thereby increase vulnerability to addiction to cocaine. This is one of the reasons why we often see occurrence of abuse of drugs, in particular nicotine and cocaine, that exposure of nicotine may actually increase somebody's risk for developing an addiction to cocaine. So that may actually be something we really want to focus some of our targeting on nicotine, because that's actually resulting in increased increased risk for cocaine addiction. Brain-derived neurotrophin factor and CDK5 are both upregulated after cocaine administration. CDK5 causes long-term desensitization in dopamine pathways, meaning that it requires greater stimulation to get the same result out of those dopamine pathways. As a result, people feel down, less energetic, less euphoric in general, and so they need more drugs to get back to that kind of feeling. Brain-derived neurotrophin factor, of course, contributes to neuroplasticity associated with self-regulation and relapse behavior. So these are both, alteration in both of these is important to understanding ongoing drug use and abuse. Similarly, opioids cause some alterations in genetic expression. In particular, we'll talk about the OPRM1 gene. So the mu-type receptor is coded by the gene OPRM1, which is highly polymorphic. We'll talk very briefly at the end today about polymorphisms in this gene. We don't have a clear understanding of how those polymorphisms are associated with drug abuse, but we do know there is something going on there. The A118G SNP is the most extensively studied polymorphism of the OPRM1 gene, which is associated with a 10-fold reduction in receptors. Research here, however, is contradictory regarding risk for opioid dependence, but there is a stronger link with alcohol. We do know that DNA methylation of OPRM1 is greater for former heroin addicts. What's really interesting to me about this is the degree of methylation differs across ethnicity. It's lower for African Americans and higher for Hispanic former addicts as compared to their Caucasian counterparts. So if we take a look at opioid overdose deaths by ethnicity, it's not clear how this alteration in methylation from heroin use is associated with risk for death. But you can see here, if you look back at the 2000 data, older non-Hispanic black addicts were sort of at the highest risk for overdose. What's interesting is there's no effective age in terms of risk for opioid overdose death in Hispanic participants. Much higher risk as you get older in non-Hispanic black participants, and much lower risk as you get older in non-Hispanic white heroin users. So there may be something going on here. The research isn't specific enough yet. There are some startling things about this. We've talked about this already, this insane increase in opioid overdose for non-Hispanic whites age 18 to 44. Dramatic increases across the board, and this really dramatic increase in those 45 to 64 in the non-Hispanic black group. So pretty interesting results here, and something we'll want to think about. So one of the things we do know is that there are transgenerational effects of opioid exposure that have been demonstrated in rats, and it's something we really need to get a handle on in understanding how use of drugs like heroin and other opioids can affect offspring throughout their lifetime, and we simply don't have the data yet. We just this is so early in this research that we just don't have a good idea about how that's affecting those offspring. Alcohol is another area in which there is some evidence for epigenetic alterations. Both alcohol use and alcohol detoxification are associated with epigenetic alterations. Chronic alcohol use may cause long-term epigenetic alterations, which again may be passed on to subsequent generations. What's one of the interesting things about this is that there is an epigenetic alteration during alcohol detoxification, and we really don't have a good handle on what's the long-term effects of that are. So the take-home message about epigenetics is we have a long way to go to fully understand how drug use and abuse cause epigenetic alterations. More importantly, we are a really long way from understanding what these changes mean in terms of behavior. Finally, we cannot ignore the transgenerational effects of drug abuse. We may be setting up future generations if we do not get a handle on this issue. So this is something that's really important, and there's a lot more work that needs to be done in this area. So I want to wrap up our discussion today by talking a little bit about pharmacogenetics and psychopharmacology. In specific, I want to talk about drug metabolism. I'm going to very briefly talk about receptor genes, but spend most of my time talking about drug metabolism because it seems to be where a great deal of what we really know affects outcomes is in that particular area. So the first thing to understand is that we're usually talking about variants of the cytochrome P genes. In particular, we're usually talking about cytochrome P2D6. They talk about, however, different phenotypes, and there are tests that are commercially available to test for these. In particular, for the cytochrome P2D6, 3A4, and some of the other cytochrome P enzymes. And they talk about these in terms of 4 different groups of people. Poor metabolizers, which seem to be having lower levels of these particular enzymes, if not missing them altogether. Intermediate metabolizers and extensive metabolizers, which are sort of in the in-between range, and the ultra-rapid metabolizers. Most of the discussion is on the poor metabolizers and the ultra-rapid metabolizers. These cytochrome P2D6 phenotypes are really highly associated with how codeine affects people who take it. Cytochrome P2D6 metabolizes about 10% of codeine into morphine. This is where most of the analgesia comes from taking codeine, is this first-pass metabolism into morphine. What the research shows is that poor metabolizers experience little analgesia. That is, they are unable to convert codeine into morphine because they don't metabolize well using this particular enzyme, so they don't get any of the morphine. On the other end of the scale are ultra-rapid metabolizers. They have much higher incidences of side effects and are at risk for a number of issues. So oftentimes they'll report nausea and vomiting from taking these drugs, and there is a potential for what's called fatal neonatal opioid toxicity. And what happens in this is our women who are given codeine, who are ultra-rapid metabolizers, convert so much of that to morphine that it is then expressed in their breast milk and then passed on to their children, and you can end up with an opioid overdose in newborn infants because of that high dose of morphine contained in breast milk. So this is one of those areas where some genetic testing is certainly appropriate before giving birth to determine whether or not you're an ultra-rapid metabolizer. So the next drug in this class that is also affected by the cytochrome P enzymes is oxycodone. It's similar to codeine. You get increased analgesia found in ultra-rapid metabolizers. The evidence is mixed about side effects and whether or not poor metabolizers experience less analgesia. Tramadol is a drug used for treating neuropathic pain. Poor metabolizers report using more for pain, and ultra-rapid metabolizers report significantly greater side effects. Again, things like nausea and vomiting. To give you a kind of a quick summary of how this works, if you look at the top, we have codeine is metabolized via cytochrome P2D6 into morphine, at least part of it is, and then that's then broken down into other metabolites by UGT2B7. Oxycodone is methylated into oxymorphone, which is a zacto metabolite by cytochrome P2D6. And it's metabolized by cytochrome P3A4 and 3A5 into noroxycodone. And then again, 2D6 and 3A4 are alternatively involved in converting that to noroxymorphone. And then finally, you can see down at the bottom, tramadol is metabolized via cytochrome P2D6, 2B6, and 3A4. I want to finish up by talking a little bit about receptor genes. There is a great deal of work going on in this area, in particular in alterations in dopamine receptor genes, in the OPRM1 gene, which is an opioid receptor. There is also, finally, evidence that melanocortin genes are involved either in receptor density or drug metabolism and have pretty significant effects on drug efficacy. So to give you an example, melanocortin is associated with hair color, and individuals who have red hair require much greater levels of anesthesia and oftentimes report greater anxiety, particularly at things like dental pain. One of the things that redheads and people who have any part of that melanocortin gene, they usually need higher levels of lidocaine and other topical anesthetics. The reasons for that have not yet been elucidated. It's either some combination of increased metabolism or lower receptor density, but they are less effective in those groups, and as a result, oftentimes people are more afraid to go to the dentist because they're more likely to experience pain. So there's a lot of work going on in this area. It's certainly something to keep an eye on and something to be mindful of. If you're really interested, you can, of course, order your genetic results from 23andMe or one of those companies. So hopefully you found this interesting. There'll be lots more coming out on pharmacogenetics, and I'll be updating this lecture throughout the year. Thanks, and have a great day.